Good evening, Pratham. Okay. So I'm Rahul Saha, and today do you want me to start you can say, errors in cell division, or shall we continue with the biological transformations? Shall we continue with the Krebs cycle or the electron transport chain, or shall we switch to the biological, this one, errors in cell division? Yes, Samesh was saying errors in cell division, I think, is in his half-yearly course. Is it in your half-yearly course too, or something like that? You don't have any exams. Okay, that's good. Okay. Let Samesh join in, then I'll ask him what else. Just hold on a second. Okay, for his sake, let's start errors of cell division. He was telling me, texting me that it's apparently given in his syllabus, like half-yearly syllabus. So let's complete that chapter first, because we have completed till the Krebs cycle. So let's start with this one. So I hope you are acquainted with mitosis and meiosis. You might have been taught that. Okay, so whatever errors can happen, like there are various kinds of errors, non-disjunction. So then you might have read about translocation. Okay, so we'll go through all those kinds of errors, what all errors can creep in mitosis and meiosis. Okay, so we have various types of errors. Okay, so in mitosis errors are quite rare. Okay, but the thing is, the process can go wrong. The process can go wrong in mitosis too. Okay, especially during the cleavage divisions, like when the zygote divides. Okay, during the cleavage, in the morula formation, in the blastula formation, and at those point in time, the process can go awry. Okay, can lead to various cell division, you can say defects. Okay, so you can have various types of mitotic errors also. These errors could be there in meiosis too, but mitotic errors could also be there. Like you may have deletion. Okay, you may have gene duplication. Okay, then you may have translocation. Okay, that you can have easily. Then you may have non-disjunction. Then you can also have inversion. Okay, so many types of errors you can have. So let's deal with these types one by one. Let's first focus on what deletion is. Okay, so what happens is sometimes a part of the chromosome, okay, which is taking part in mitosis, can get damaged. Okay, it can get damaged, and what can happen is it will lead to certain deleterious events, meaning that there may be some breaks. Suppose this is the chromosome. Okay, this is the homologue of that one. So there may be some breaks induced in this chromosome. Suppose a break happened and this part went off. Okay, so what happens is, if the chromosome gets broken, what would happen is the fragments which were broken off during the mitosis or mitotic division, those specific fragments can get lost after the mitotic division is over. Okay, so the thing is, when this fragment is lost, it may contain some genes too, right? Even though you have 98.2% as the heterochromatin region, which we have talked about in detail regarding the histone octamer. Okay, and 1.8% is the genic region or genes. Okay, euchromatin. then also you can lose some genes from here too, and that may lead to excessive, you can say, phenotypic alterations in the cell, or in the cell which has been divided and which has become now new cell, right? So that specific cell will not be having those specific genes which were already present in its predecessor cell. So like that you can compare. So that does lead to deletion. This is an event known as, or this is an event known as deletion event. Okay, so that can also happen sometime. It is called deletion. So you may have a part of the chromosome which may become truncated and left off. Okay, so that is called as deletion. Good evening, Samanish. So have started your specific topic of liking errors in cell division. Okay, so then coming on to translocation. In case of translocation, it is little bit different. When you talk about translocation, so the chromosome in case of translocation does not have to be homologs. Okay, homologous chromosomes are not required here, or are not the participants. Are not the participants. Can be the participants, but are not really the, you can say, mandatory participants when it comes to translocation. In some rare events, it can happen between the homologous chromosomes too. Okay, but the thing is, this translocation process is what? You are translocating from one place to the other place. Okay, that is known as translocation. So when you are going for translocation, what you are really imagining, what you are really able to see, what you are really able to confront is, you can swap specific parts of one chromosome with other parts of any other chromosome, not its homolog, or not its homologous chromosome. That is the meaning of translocation. Because swapping of chromosomal material, swapping of the, you can say, genetic data, genetic element between the two different chromosomes of different chronological number. That is the meaning of, you can say, translocation, just breaking and reattachment. So you can have various types of translocation. Okay, one can have translocation in this pattern. Suppose this is chromosome number 9, this is chromosome number 22, because 9 will be bigger than 22 because of the chronological order in which they are arranged. Okay, so what can happen is, there is something known as Philadelphia chromosome. Philadelphia chromosome. So translocations could be reciprocal, non-reciprocal, Robertsonian. So we'll talk about the reciprocal translocation here. What happens is, this contains BCR gene and this contains ABL gene. Okay. Promoter is there. Now a part of this ABL gene gets truncated and gets attached here. Some part of this gets truncated and gets attached here. Okay. So the result is, chromosome number 22 now will contain BCR region. Downstream of that, you will be containing the ABL region. Okay. BCR is breakpoint cluster region, and ABL is a specific tyrosine kinase. Okay. ABL is nothing but a tyrosine kinase. Okay. So tyrosine kinases are the enzymes which transfer the phosphate group from ATP. Okay. ADP plus PI, this phosphate group will be transferred from ATP to PI to the target molecule's tyrosine amino acid, to the tyrosine residue of the target molecule. The target molecule's tyrosine will be phosphorylated. Okay, will be phosphorylated. That is done by kinases. Kinases come under the category of transferases. We have 6 categories of enzymes I taught you. The kinases come under the category of transferases. They are transferring the phosphate group to the tyrosine residue of the target protein. Okay. or you can say tyrosine amino acid. So tyrosine is an aromatic amino acid, tyrosine, phenylalanine, and tryptophan. So what happens is when the ABL gene was present here, okay, no specific cellular division, you can say haywire or haphazardness went on. Means the cellular division went on at a specific rate. Okay, the cycling CDK kinases, they were all, you can say, following a specific, you can say, moderate pathway. They were not highly upregulated or downregulated. The checkpoints, G1S checkpoint, G2M checkpoints were, you can say, surpassed at an appropriate rate. But what happens in when this ABL gene gets switched over or it swaps position and it's then relocated, or I can say translocated to the proximal part of the BCR, means it is pretty near to the BCR gene now. Now what happens is BCR, this breakpoint cluster region, it acts as a very strong promoter. Okay, the promoters can recruit RNA polymerase enzyme. So when I'll teach you in biotechnology only later on, what happens is for any gene to be transcribed, okay, if you take any specific gene, only the gene is never enough to get you protein. What do you need? You need, this is a DNA, right? The DNA cannot come out of the nucleus and bind to the ribosome present in the ER or of endoplasmic reticulum. So what happens is, what is needed is, you have a specific region upstream, means before the gene, known as the promoter sequence. Okay. So the stronger the promoter sequence is, the better it will be able to recruit the RNA polymerase enzyme. In eukaryotes, it recruits various, you can say, RNA polymerase binding proteins, okay, or transcription factors they are known as, especially for RNA polymerase 2, which is present in eukaryotes. But in simply, or in very, very simple terms, you can think of that this will recruit RNA polymerase, and then a complementary messenger RNA will be produced, mRNA, which will then be exported out of the nucleus, and that will bind to the ribosome, mRNA. And unless mRNA binds to the ribosome, the translation process or the protein synthesis process, that is known as translation, okay, it cannot begin. Then only it begins or commences. And this process, the conversion of DNA to mRNA or formation of mRNA by analyzing the DNA by using template DNA, okay, DNA as a template, is known as transcription. Okay. So what happens is this is specific. So this specific mRNA is produced from the, or due to the promoter. So what happens is, if this becomes BCR and the gene is the ABL gene here, so now this BCR is a very, very strong promoter, you can say. And this will cause overexpression of ABL gene, means more and more mRNA will be produced. More and more mRNA means more and more of ABL protein will be produced. More and more ABL protein will be produced. If more and more ABL protein is produced, what do I mean then? Means the cell cycle, or the cell cycle speed of the cell cycle could become very, very high. Means the cell can divide at an accelerated pace now, which is a hallmark of cancer. Okay, so now you can get cancer. That is known as specialized type of cancer, specialized type of blood cancer that you will be getting is chronic myeloid leukemia. Okay. So when you have a hematopoietic stem cell, you have the lymphoid precursor, you have the myeloid precursor. Okay, this is known as hematopoiesis. You can have acute myeloid leukemia, chronic myeloid leukemia. Okay, you can have acute lymphocytic or lymphoid leukemia, okay, or chronic, you can say lymphoid leukemia. Then you may have Hodgkin's lymphoma and so on. Okay, various types of blood cancers you could be having, right? Rocky Balboa's home. Yeah, that is there. You are correct. Good evening, Arya. That is correct. Rocky Balboa, Sylvester Stallone, okay, Philadelphia in US. You are absolutely correct. Okay. So the thing is, there could be many types of blood cancers, but this chronic myeloid leukemia, okay, this one, CML, is caused by a translocation event which can happen in our cell, okay, and that could lead to, if that happens in the WBCs, that could lead to blood cancer known as chronic myeloid leukemia. So that is our Philadelphia chromosome. Okay, so hope that it doesn't happen to anyone, but there is a possibility of that happening too. Okay. Now, once we talk about, this is a reciprocal translocation, like that non-reciprocal translocation can also happen. Non-reciprocal translocation can also occur. Now, in case of non-reciprocal translocation, what happens is, suppose the gene gets swapped from here, or a part of the chromosome gets swapped from here and gets attached here only. There is no reciprocation from this end. Okay, there is no reciprocation from the other chromosome. So this is chromosome number 10, this is chromosome number 11. There is no reciprocation from the other end. That is known as non-reciprocal translocation, means from one end only the chromosome, or from one chromosome only the gene will be swapped. Know it very very clearly, from one end only the gene will be swapped, not any other. Okay, so that is known as, the result will be this chromosome becoming longer and this chromosome becoming even shorter. Okay, because it has got now this specific piece here attached. That is known as non-reciprocal translocation. Now, coming on to Robertsonian translocation, it happens in very very, you can say, rare one out of 1000 babies it occurs. Okay, Robertsonian translocation. One in 1000 babies you can think of. Now what happens is, this happens in acrocentric type of chromosomes. Very very important. Acrocentric chromosomes. Now acrocentric have one end quite small and other end quite big, right? Just before the telocentric one. Metacentric, submetacentric, acrocentric, then telocentric. Now, acrocentric chromosomes are which chromosomes? There are 5 pairs of homologous chromosomes which are acrocentric in nature in the human genome. You have 13, 14, 15. Chromosome number 13, 14, 15, then 21 and 22. These specific pairs of chromosomes are known as acrocentric chromosomes. Chromosomes in the human genome. Okay. So these are the acrocentric chromosomes present in the human genome, right? Now, once you have this acrocentric chromosome going on, okay, which is the meaning that these are present. So what can happen is, there could be translocation between these two or any two acrocentric chromosomes. Even between the homologs it can happen, but between any two acrocentric chromosomes, the translocation could take place. And once it does take place, what happens is, this is a very very special event. Okay, just look at this, how it occurs. So what happens is, you take one acrocentric chromosome. Okay, suppose you took the chromosome number 13. Now you took the chromosome number 14. what will happen is the larger fragments known as the QUE or fragment Q, this is a smaller arm, petite or P, right? So P arm, Q arm. So what happens is the smaller the P arm is extremely small, or you can say smallest in case of the acrocentric chromosome. So what happens is the exchange occurs only between the two P arms. Okay, the exchange occurs only between the two P arms of the two acrocentric participating chromosomes. Now, what could be the result? The result will be the two P arms they go together and the two Q arms they go together. Okay, this is a specialized reciprocal translocation. So it will result in one huge chromosome because two Q arms have got attached. So one huge chromosome and two P arms have got attached. So one a very redundant or truncated chromosome. Here both the P arms have attached. Here both the Q arms have attached. Now, one more speculation or not speculation, one more research data here. The thing is, where do you see more genes? Here or here? So, the majority of the genes will be on the larger arm because the P arm is really petite, really small in case of an acrocentric chromosome. So, since it is so minuscule in an acrocentric chromosome, what we will have is the genetic content, okay, the genetic data, the number of genes will maximally present. The probability of those genes to be present at maximum will be rendered upon the larger subunit or the larger, you can say, fragment, the Q fragment, the larger arm of the acrocentric chromosome. What happens is there is a very less or no net loss of genes. Okay, the genes are not lost. Mostly they are not lost. Some genes could be here, but not. The likelihood of the genes present in the really small P arm is quite low, quite less. Okay, so the genes are not lost. You can say in 90% of the cases. So the genes are all recovered, but this chromosome is extremely small, okay, and made up of almost no genes. So what happens is in the further divisions, it cannot undergo any kind of homologous, you can say, recombination because it is so small. There is no homology between this one and any other chromosome. So it ultimately gets completely eliminated from the cell because it cannot take part in homologous recombination. Same is the theory for the human Y chromosome. If you think, if you have read any research paper, you would be thinking that I'll just show you the karyotype of the human chromosome. Karyotype, human chromosome karyotype. Karyotype. Okay, so just look at any one of these images. Okay. This is a good image. Just look at this image. Okay, look at X and Y. Okay, the Y chromosome is so small in front of the X chromosome. You see this Y chromosome is so small in front of the X chromosome. So earlier, millions of years ago, the Y chromosome was not that small. Okay, it was quite large, but now it has become truncated. Why? Because it doesn't have any homologous chromosome recombination partner, no partner with which it can undergo homologous chromosome and get acquire variation, acquire the chromosomal segments. Okay, so what happens is that the two X chromosomes, they have X and X, they have the homologous pair to undergo recombination, but Y is all alone. It cannot undergo homologous recombination because in homologous recombination you need homology, you need gene similarity, gene identical genes, similar genes, so that they can undergo homologous recombination via propagation or formation of holiday junction. That is not in your course. Holiday junction and you can say branch point migration, resolution of the branch point, those are not in your course. Then RUV, C proteins, REC BCD, okay. In humans, RAD, you can say RAD 51, SPO11, these kinds of proteins that come and take part. The thing that I'm trying to convey here is you don't have any partner for Y chromosome. so that homologous recombination or simply crossing over cannot occur. Okay, so it has become redundant in size little by little, and it is speculated by scientists that after a million years we may lose Y chromosome completely. Okay, so there may be no maleness remaining. Okay, once you lose the complete Y chromosome, there may be no maleness or male remaining, because the male is dependent on being Y chromosome or having the Y chromosome, so it can be lost completely. So after a million years, there may not be any more males remaining. That could be the end of the human race. Okay, that could be definitely the end of the human race, because the males may be completely eradicated. Okay, by that time, if the evolution does keep on taking place and if you have proper natural selection, then what can happen is some of the genes which are required for the sexual characteristics of a male present on Y chromosome, they can translocate to any other chromosome. That can happen. Okay, and then that specific chromosome can become the carrier of the male characteristics. That is only one way, but again that will have major complications regarding the determination of the sex. Okay, so still we have to wait and watch the entire, you can say, sequence or saga of evolution for the next 1 million years. Okay, nothing can be done about this. We cannot just put one more chromosome. Okay, even if you put, it will cause aneuploidy. Okay, so that is a great problem that especially the males are facing. Okay, so that is known as, so that is why this smaller chromosome will be eliminated. Okay, it won't have any partner. This bigger chromosome, okay, will stay, and then this is known as, and this will lead to then aneuploidy, one less chromosome. This is known as Robertsonian translocation. Okay, very rare event, but a prominent event known as Robertsonian translocation. Next event in this case scenario only is the inversion. Okay, genetic inversion event. Again, you have chromosome. Okay. So there was a research 2 days ago. The way we draw chromosomes like this, right? We draw chromosome like this: centromere, C, and P, A, and all. You can say specialized centromere. S3 histones are present, so it is known as centromere. Then you have chromosomal, you can say, passenger complex, CPC, located in the centromere. Okay, kinetochore. So all these specific diagrams are incorrect. Okay, so I'll send the research paper to you. Personally, you can go through it. I'll send the latest picture of the chromosome also. Okay, so the which has been captured by high resolution microscopy. So I'll send it to you live images. Those are in the live cells, not in the dead cell. Okay, such great 3D images. Okay, in US the research was conducted in. So great research. Okay, and the thing is, this is this is known as inversion event. In inversion is, you can say, suppose this is a gene. Okay, if you just amplify the gene. Okay, so let's stick to this model for now. Okay, of chromosome, but this is not any more correct. Okay, as per the research which was published 2 days ago only. Okay, so once we amplify this gene, let's give it an, or attach it an arbitrary sequence. Okay, let's put an arbitrary sequence to it. So suppose A, T, G, C, C, G, A. This is the sequence. Okay, and the complementary will be T, A, C, G, G, C, T. This is 5 prime to 3 prime, 3 prime to 5 prime. Now, once inversion happens, inversion means flipping. It is like this. Inversion means like this. Flipping will happen. So the entire gene will twist. The entire sequence will invert. Okay. The sequence could be just the opposite now. Now it can run from like this: A, G, C, C, G, T, A. It could be like this here: T, C, G, G, C, A, T. Now, what could be the ramifications here? The ramifications could be severe. Okay, in class 12 we'll be studying about genetic code. Okay, so this is an event which is related to genetic code regarding its application. Meaning that, just understand one thing. I'll tell you, I'll try to explain it to you in brief. The genetic code event, it is given in detail in class 12th. Okay, what happens is, what does a gene code for? We all say it codes for proteins. How does it code for protein? Have you ever thought of that? So it codes for protein by coding individually for amino acids, and amino acids have a specific sequence in a protein. How does insulin differ from glucagon? Both are proteins, but the sequence of amino acids of insulin is different to that of the sequence of amino acids present in glucagon. Okay, and then there could be multiple polypeptide, single, you can say polypeptide chain, that is different. You can have multimeric, monomeric, but suppose two monomeric proteins are kept, means two single polypeptide comprising proteins are present. How will you know, how will you discern, how will you recognize the difference between the two polypeptides? That these are two different proteins altogether. By knowing their sequence. Just like your and mine genetic sequence, they may differ at the microsatellite region, millisatellite region, thereby I am not your father, you are not my son. It won't match. Same way, the two protein sequences, if they match completely, they are the same proteins. If they are different, by their or when their sequencing is being done, the result is the two protein sequences are completely different. Okay, not one or two regions of polymorphism, completely different. That means they are not polymorphic proteins, they are completely different proteins. So what happens is, suppose the thing is, you divide the genetic or the gene sequence, the DNA sequence, into a specific group of three codons that are known as codons. Okay, so three grouping is done like this. Suppose you have a T here, so three grouping. Okay, it will become A. So group of three. Wait, 1, 2, 3, then 1, 2, 3. Like this, it will happen. 1, 2, 3, 4, 6, 7, 8, 9. So put C here. So let's make it group of 3 for you to understand properly. The group of 3. So here E, A, C, G, E, C, E, E, G. So here 3, 3, like this. So what happens is, the mRNA which is coded, which will be the, this will be the template, so this will be the mRNA. So from mRNA, three nucleotides will be present, and that will act as codon. Okay, so each of the three pair or each of the three group may code for a different amino acid. This may code for glycine, assumption only. This may code for leucine. This may code for valine. So the polypeptide coded by this segment, okay, we'll have glycine at the first position, we'll have leucine at the second position, we'll have valine at the third position, like that. Okay, now if we have reversed the chain or we have reversed the sequence, what could happen? First, second, third. Now the first position will go to valine. Okay, it reversed it just like this. Second position went to leucine only, but third position went to glycine. Okay, like this it can happen. The ramification could be like this. So the entire polypeptide sequence may differ. You have the same amino acids, but the sequence differ. Once the sequence of chronological order differs, the entire protein becomes something else. And when it becomes something else, will it attain the same structural conformation or conformation stability? No, it may not bind to its substrate if it was an enzyme. It may not bind to its receptor if it was a hormone, whether it was a specific, you can say, hormone which is water soluble or lipid, means it binds to the receptor. If it was a water soluble hormone, it would bind to the receptor present on the plasma membrane. If it was a lipid soluble hormone, it would just go through the plasma membrane and bind directly to the nuclear receptor. It may not bind to any, right, and it may not then perform its proper function, and you may have again a phenotypic ailment, a disease, right? So this is the problem with inversion. And why you take three amino acids or three DNA sequences 3 nucleotides for one codon is, the reason of that also I'll tell you. The reason is, how many nucleotides do we have? 4, and there are 20 essential amino acids. Okay, 20 essential amino acids. Now, 4 to the power 1, if only one nucleotide could code for the amino acid, then 4 to the power 1 will give me 4. So I could only code for 4 amino acids. Means I'm still lacking 16 amino acids, which cannot be the case. Then I would be dying, right? Then 4 to the power 2. So 4 into 4, 16. Now I'm coding for 16 amino acids, still 4 amino acids are left. Again I'll die. Now 4 to the power 3, that is 4 into 4 into 4, that is 64. Fine. So now I have been able to code for 20, but 64 means many other codons could code for the same amino acid. Okay, but at least 20 will be coded. I will not code for 25 or 30 amino acids. What will happen is it may generate redundancy. Okay, or you can call it by a better word, degeneracy. Means multiple codons. Okay, multiple codons can code for the same amino acid in a genetic code, or in our body, in bacterial cell, anywhere. It is a universal genetic code. Okay, suppose AUG codes for methionine in humans, AUG also codes for methionine in bacteria also. Okay, so that is the thing, it is universal. So multiple codons can code for the same amino acid, but it is also ambiguous in nature. Ambiguous means one gene, sorry, one codon cannot code for more than one amino acid. That must be very, very clear. Cannot code for more than one amino acid. So this will become very clear when I show you the genetic code here, okay, of humans or of anyone. That is a universal code, genetic code table. Just look here. What do I mean by that? You can see these four codons CUU, CUC, CUA, CUG, they code for leucine. If you check serine, check ACR, that is serine, another amino acid, AGU, AGC2, right? And then 4, UCU, UCC, UCA, UCG, because this is from mRNA, that's why T has been replaced, thymine has been replaced with uracil. Okay, so this is as per the mRNA. So how many codons are coding for serine? 6 codons. Okay, methionine and tryptophan, if you see, okay, so UGG is coding for tryptophan. You won't see any other codon coding for tryptophan. Okay. Same way, if you check methionine here, okay, so methionine is also coded by only one AUG, only one codon. So only methionine and tryptophan are the two amino acids which are coded by only one codon. There are no, you can say, multiple codons for their coding. But if you add this, you can say 4 plus 4 plus 4. If you keep on adding, you will get 64. These are the 64 combinations of the codons which can be present in a triplet form in the human DNA, which is converted to mRNA. Okay, so that is the genetic code, and the entire sequence of amino acids is deduced from this code only. Okay, which is present in your DNA sequence directly, which 333 you can segregate, group 333 nucleotides or bases, and then you will be able to form the amino acid. Okay, so inversion will cause flippage, or you can say direct reversal of the amino acid sequence. So that is why inversions are very, very dangerous, you can say, event. Now there are two types of inversions. One is paracentric one is pericentric. Pericentric inversion. Yeah, that is true. Females would be more, males would be very less. Okay. So in case of paracentric inversion, what could happen is there is only one cut in the chromosome. Okay, so that won't involve centromere. No centromere is involved. Okay, centromere is not involved in the paracentric inversion. And moreover, there will be only one cut. The chromosome can be reversed and can be ligated again. Okay, that is your paracentric inversion. In pericentric inversion, one cut in only one of the arm, either the P arm or the Q arm, will be present in paracentric inversion. When it comes to pericentric inversion, you will have two separate cuts, one in the P arm, one in the Q arm, petite and Q arm, okay, longer arm and the shorter arm P arm and the longer arm Q arm, okay. Both the cases you will be having, in both arms you will be having a specific cut, okay, a specific excision, and then this entire region would be flipped, okay, would have opposite orientation, okay, the orientation would be reversed completely, and then what you will be having is this new pattern where this has been flipped, okay, and centromere was involved. That is known as pericentric inversion, involvement of centromere. So both these events can occur. Same way gene duplication can happen, pseudogenes can be formed, okay. Sometimes the replication enzyme, there is a loop in the DNA, it can cause double replication of this one. Straight away, it can read it again, so it can lead to gene duplication event. Okay. Sometimes there could be an under loop in the DNA, it could directly go through, it could miss. Okay, that can also lead to gene deletion event. Okay, then there are something known as pseudogenes, okay, which may be having some stop codon, but their other sequences, they look like the genes. Okay, so there are pseudogenes too in the human genome. Okay, there are transposons in the human genome that we have talked about earlier. Okay, so those things we have talked about. Then we have talked about, we'll also talk about non-disjunction. Okay, so non-disjunction is a specific event where you are not disjoining, where you are not disaggregating. That is the meaning of non-disjunction. It happens mostly in the meiosis. Okay, in the meiotic event it happens. So we know that in meiosis the first event is of prophase 1. Now, prophase 1 contains pre-leptotin, then leptotin, then zygotine, then pachytine, then early diplotin, then late diplotin, then you have dikinesis. So, in the zygotine stage, you have something known as synaptonymal complex formation. And when that synaptonymal complex forms, there are multiple proteins, there are mini chromosome maintenance proteins, okay, DNA helicases. Then you have structural maintenance of chromosome proteins. These are the main proteins which come into effect when you have synaptonymal complex formation, SMC proteins. Okay, so that means the centromere of the two homologous chromosomes, okay, are connected in a way by the synaptonymal complex. This is the synaptonymal complex, okay, SC. Now, see, after synaptonymal complex formation, you have chiasma formation, chiasmata formation, okay. That is the meaning of the advent of, you can say, crossing over. That happens in the pachytene stage. Now, after crossing over happens, you have Holiday junction formation, RUBC, REC-BCD in eukaryotes, RAD, 51, SPO11 pathway. Okay, those kinds of enzymes come in. You have branch migration. Okay, you have Holiday junction resolution. So that detail is not given in your course. So what happens is you have crossing over event. Once you go into the crossing over event, suppose you have this gene, that is there, and then we have something known as now when they cross over means they cross over, so this could cross over like this could cross over like this after chiasmata formation. So what you will get is B. Now A has crossed over with small b, so capital A, but this side will be your small b. Okay. Same way this side small a, small b. Now B has crossed over with small a. Okay. So this side will be small a, but this side will be capital B. So how many percentage of, or what is the percentage of recombination here? The percentage of recombination, even if the recombination between these genes happen for 100% of the times, which is never the case between these two genes, recombination never happens 100% of the events. Okay, between the two specific genes, it could happen between different genes also. But if we consider these two genes in the homologous chromosome, and recombination happens 100% of the time between these two genes only, A and B, still 50% are only the recombinants. You can see these distal chromosomes are still the parental type chromosome. They are the recombinant chromosome. So maximum recombination frequency is reached. That is again only 50%. After this recombination event, genetic recombination, when you produce a lot of variation, right? What we have is we have resolution. We have dissolution of the synaptonemal complex. And when that dissolution at diploting stage, early and late diploting stage, it cannot happen properly. What you have is improper dissolution, and that could rapidly lead to the dissolution is not happening properly. Okay. Suppose it is like this, that dissolution is not happening properly. Okay. So one cell could go with three chromosomes. Okay, one, two, one more. Okay. So it could go with three chromosomes. Okay, it could go with three chromosomes. The other cell will have only one chromosome. So once it goes with three chromosomes, other cell goes with one chromosome. So the total number of chromosomes in this cell increases by a number of one, and this one decreases by a number of one. So what does this make? This changes the ploidy of the chromosome. Ploidy means the number of chromosomes which are supposed to be present in one cell. So euploidy means the true ploidy of the chromosome. If the number of chromosomes in humans are 46 in diploid, that means it is euploidic in nature. But if this event has happened, then what will happen? One extra chromosome. That is the meaning of aneuploidy. So U means true, ploidy means number, and means false. Okay, ploidy means number. So aneuploidy, so false. The U has become and, so false ploidy means you are getting here 47, sometimes 48, 49, sometimes 45 chromosomes. Okay, so the chromosome number is not as per what it should be, and not as per the designated chromosome number present in the specific species. So each species has a specific designated chromosome number. If you differ from that, if you go haywire from that, if you do not comply to that specific criterion, then what you generate is aneuploidic event, and that could be very very deleterious because we depend on the gene, you can say, products, that is the protein. So if you have three chromosome 21, so suppose that chromosome number 21 trisomy happened, this 3 happened at the chromosome number 21st pair, that will be then known as Down syndrome. Okay, just an example. So here you have three chromosomes, so this will be transcribed, this will be transcribed, and this will also be transcribed. So you may have excessive transcription of the genes that can also hyperactivate the immune response, cause retardation, mental retardation, protruding cheek, then ultimately early death. So there was a latest research paper which showed that the interferon receptors okay, which are the proteins produced after viral infection. Interferon gamma receptors are produced on, are present on, that gene is present on chromosome number 21. So that gene is hyper expressed because you have 3 copies. So more and more interferon receptor, okay, is produced in the patients suffering from Down syndrome, and that will hyperactivate their immune response. That can also happen. And in the cell where only 45 is there, so the gene concentration, the product concentration may be lesser. Okay, that again you will be suffering from that less gene product ramification, right? So the things, they keep on getting worse and worse, right? So I hope you are able to comprehend this lecture. So in the next lecture, we will talk about more about euploidy, aneuploidy, polyploidy, okay, chromosomal defects, point mutations, and all such events which are the part of the integral part of the errors in the cell division. Okay. So if you have had any kind of doubts or queries, you can definitely clarify it on my personal number. I'll reply to you as soon as possible. Okay then, thanks a lot. Let's